All right. So I'm doing my dissertation on low-cost nutrient reduction techniques and their impacts on nitrogen cycling and microbial communities. Why do we care about nutrient reduction techniques? Because nutrient pollution is such a persistent issue across the United States, but especially here on the Gulf Coast. So we have two major sources of nutrient contributions to streams, which are fertilizers and sewage. So whether it's an agricultural field or a leaky sewer pipe, we can have leaching of nutrients that then enter catchments. And once we're in catchments, or even on our way to catchments, one of three things is going to happen. They can be advected downstream and enter estuaries. They can enter dissolved storage, so they hit the subsurface and then hit groundwater, where they can be stored for months to even decades before entering catchments. Or they can hit particulate storage, so they can be assimilated by biomass or absorbed onto particles. Now, to put this into context, over 50% of Alabama, the majority of Alabama, drains down to Mobile Bay. You can see on the right-hand side of the screen right there, we have the state of Alabama, and all those yellow areas are ephemeral streams. So small little catchments that drain to larger areas for only a fraction of a year. The blue are persistent catchments. So a lot of those are going to be first or second order streams. And of those first and second order streams and ephemeral streams, we have over 130,000 miles of stream length that contributes to the Tombigbee and Alabama rivers. And those can contribute over 40% of the total nitrogen loads that eventually hit Mobile Bay. And that impacts us in a really tangible way because we've correlated nitrogen loads with phytoplankton blooms in the upper bay, which can have negative impacts on economically important species like blue crab. Now, if we want to target this to export nitrogen before it actually hits these catchments, these larger systems, how would we do this? Well, denitrification is a fairly simple mechanism in terms of remediation strategy. So it's an anaerobic form of metabolism that can take nitrate, the most abundant form of nitrogen in plant fertilizer, and transform that into N2 gas, which can then flux out of the system. However, it requires suboxic conditions. So if we're trying to remediate a stream, we can't really accomplish this in the water column because think about your remediation area. You're constantly having fresh water, which is flushed with oxygen, move through that bin. So we need to target the sediment. The sediment is a much more stable condition where we have longer residence times. But in order to actually draw down those advected nutrients, like in the water column, we need to target the hyporic zone. So this is an ideal niche where we have this ecotone between the water column and the deeper subsurface that actually exchanges water and can target nutrients more effectively. But in order to actually stimulate denitrification here, we have to address a few rate controls. First is residence time. That's fairly obvious. We have long residence times. That means we actually have biochemical oxygen demand that's reaching the dissolved oxygen in that parcel of water. It can draw it down, so we actually have this anaerobic environment where we can have denitrification occurring. A second control would be lability. So it's a form of metabolism. It requires carbon. Same thing with aerobic metabolism, that also requires carbon. So not only do we need carbon reaching the denitrifiers, but we need enough labile carbon to draw down the oxygen in the first place. Then onto a more nuanced issue is the carbon to nitrogen ratio. So this goes to the stoichiometry of the overall denitrification reactions, but on a more basic level, nitrogen is an essential nutrient. All these microbes are going to need it to synthesize proteins and other essential structures. So if we don't have enough nitrogen relative to carbon, we're not going to be able to actually have this flux out of the system. Okay, they can still hear us. So if we don't have this flux out of a system because we don't have enough nitrogen, we're having assimilatory uptake. And so we're not actually fluxing anything out. So we need to be careful when we're choosing our sites and how we're introducing organic matter as a means of stimulating denitrification. So to actually implement this in a stream, we're going to be looking at shallow systems. So we want strong benthic coupling as a result of shallow water relative to the sediment, relative to that hyporic zone. We want low flows, that way we have longer residence times and we're not flushing too quickly. And we want abundant organic matter that is relatively close to the benthos, if not directly on it. And so we're proposing a organic matter filled mesh. That way we have flow through of the organic matter, but it is attached to the benthos. It can be seen as an extension of the sediment with good exchange. And to actually test this design, we're going to be implementing a Bakke experiment. So a before-after control impact experiment. We're going to use an upstream site and a downstream site and compare nutrients of the two. We're going to use a year of prior baseline monitoring and a year of monitoring while this gear is actually implemented. So for study sites, we're going to be using 3 Mile Creek as an example and different branches. So this is the South Alabama campus. In the northeast portion, we have these sandy, heavily incised forested streams. But on the western portion, we have more organic matter rich streams where we have these grasslands that can actually leach fertilizer directly into the stream. It's a mix of riffle and sandy pebble soils. And we have a more swampy site at site number 4. Now, coming back to that carbon to nitrogen ratio issue. One concern when we were setting this up is, do we actually have enough nitrogen to see drawdown in these streams? So we did a pilot experiment and we saw that we do actually have a good amount of nitrate in these systems already. So 16 micromolar is what you consider anthropogenically stressed. We have in the range of 5 to 75 micromolar of nitrate in these systems. So it's safe to say that this is a good site. Now on to my actual chapters. The first one is going to examine denitrification rates in these low order streams. One question I want to address is, how do carbon to nitrogen ratios compare to carbon lability in their relative importance for denitrification? The second is, what would be the dominant mode of metabolism in these streams? Let's say we get them anaerobic. Denitrification isn't the only nitrogen cycling pathway. We can also have dissimilatory nitrate reduction to ammonium. And this is a pathway that takes that nitrate, but instead of taking it to N2 gas, we put it and turn it into ammonium. And so it's not actually leaving the system, it's still an ion. To test this, I'm taking whole cores from the streams. I'm spiking them with 15 labeled N in the form of nitrate and adding bromine as a tracer so we can examine the pore water exchange from that overlying spiked water to the sediment to see what portion is actually getting bound to the sediment versus stuck out of there when we take this signal from a siphon. And we're also doing destructive endpoints so we can get the amount of nitrate that's also in the sediment at the very end of the experiment as well. For the treatment conditions, we're taking different forms of carbon. So we want to see what's bare sediment, what's going to be able to be cycled just from what's bound to the sediment itself versus sediment with leaf litter, which we would naturally find in these streams. Is that sufficient? Or do we need to add labile organic matter in the form of varying C to N ratios from compost? For the in situ work, we're going to be examining how this carbon lability can work as a control for the nutrient export into streams. And also, is there sufficient exchange between the hyporic and the surface waters for this to matter? So just a real basic timeline. I've gone over most of this already. The first year is baseline, second year is the actual implementation. But in the second year, I want to point out that I'm also doing microbial work, which I'll get to shortly. And I'm also doing stream gauges and groundwater sampling as a means of examining the two. So why is the text messed up? Anyway, so for the in situ experiments, I'm looking at carbon, nitrogen, and phosphorus as the major nutrients. We're looking both upstream and downstream. We're taking water, sediment, and groundwater portions. For the water portion, we're looking at the inorganic as well as the organic forms of all these nutrients, the dissolved and the particulate forms of all these nutrients, and then specifically the nitrogen species. So nitrate, nitrite, and ammonium. For sediment, we don't really care about all that. We're just looking at total portions. We want total carbon, total nitrogen, total phosphorus. And we're also logging O2 because one concern is how much will this hypoxic zone actually spread within the stream. We want to make sure we not only have this hypoxic zone at the site, but it's not propagating downstream. And then we're also taking discrete pH, temp, and PAR measurements. For the groundwater, we're looking only at the dissolved portion. So we're doing all of these same measurements for organic and inorganic fraction, but only the dissolved portion, because when we're taking groundwater, we're not expecting to have particulate transport through the sediment itself. That's going to be pretty much negligible. And once we have both the groundwater and the surface water, we can do an end-member mixing analysis. We can see what portion of a nutrient regime can be attributed to advection versus the groundwater itself. And we can develop this once we have flow rates through ultrasonic sensing of stream stage and then back calculating stream flow, as well as doing some basic Darcy's law calculations for the pumping of a groundwater. And finally, I like to think of this as looking under the hood for the whole mechanism. So we're taking microbial samples from all these other experiments, and we're seeing what exactly is happening to drive all these changes to nutrients. Or if we don't see changes to nutrients, are we seeing changes in the microbes anyway and it's getting counterbalanced somewhere? This is seeing proof of concept, more or less. So will community composition change to end cyclers? How has the gene expression changed? And will near S to near K ratios change? So near S to near K ratios can be used as a metric for complete denitrification to incomplete denitrification. Are we seeing a community shift, but for some reason we're not seeing N2 shift out? We're still retaining nitrogen in the streams. Are we perhaps seeing the wrong microbes get expressed? And to do this, we're examining this with meta-omics and amplicons. I'm not going to get into all the sequencing techniques and all the analysis, but just broadly, metagenomics is looking at all the DNA in the sample. Metatranscriptomics is looking at all the RNA. And then amplicons are looking at specific target sites. So for metagenomics, we're going to look at how the end cycling guild has changed and how the gene abundance has changed relevant to the community itself. And for metatranscriptomics, we're really looking at what functions are the community performing. We're using 16S and ITS as means to a really cost-effective way to compare a baseline for the stream. We're looking at upstream and downstream and seeing, has the community really changed on a spatial scale, or are we just static? For a sampling regime, we're looking at monthly metagenome and seasonal metatranscriptomes for these in situ experiments, and we're looking upstream and downstream for the amplicons. So the metagenome and metatranscriptome, those are all at the site itself, because we're not expecting to see much change 20, 30 meters away from this. For the cores, we're taking amplicons beforehand, because again, it's a cost-effective way and we're not expecting to see change. But the metagenome and the metatranscriptome are going to be for the endpoint sampling. That way we can actually correlate gene transcription to rates of denitrification. Now, this work is going to fill a crucial knowledge gap between practical remediation strategies and their actual microbial underpinnings. So what are we doing and what it's actually doing on a very basal level? And it's going to test the feasibility of these low-cost methods of just literally taking compost, putting it in a mesh sack, and throwing it in a stream, and seeing how that's going to work in stream water, pore water, and sediment storage, and changing those nutrient regimes in those bins. So I'd like to thank Osprey, the EPA, and Vesta, as well as my community members for all these funding.
I'm presenting my prospectus. At this point, I have pretty much collected all of the data that I'm going to use for my thesis and processed about half of it. So I have some preliminary results that I will be able to share to explain my methods and my hypotheses. All right, so for my research, I'm comparing two different types of underwater video systems that are used to collect data on fish in the Gulf of Mexico. The first camera system is called an ROV, or remotely operated vehicle. It is a robot with cameras attached to it that we drive around underwater. The second camera system is called a 360-degree baited remote underwater video system, or a 360 BRUV. It has 5 cameras that all point in different directions. Those cameras all take a video simultaneously, and that video can be stitched together to make a 360-degree video. Both of these cameras are standardly used in large fisheries surveys in the Gulf of Mexico that are funded by both states and the federal government. That includes CMAP, the Southeast Area Monitoring and Assessment Program, Alabama's Offshore Fisheries Independent Survey, Florida Fish and Wildlife Fisheries Independent Survey, the Panama City Survey, and the Greater Amberjack Count. which is a multi-million-dollar project involving dozens of institutions from Texas to North Carolina. Both of these camera gears collect data on things like the abundance of fish, the species composition of fish, and the size of fish, and that affects things for fisheries managers who are deciding season lengths, fishing quotas, size limits, or other important fisheries management decisions. It's very important to compare the data between these two camera systems to figure out how to correlate that data. So surveys that use both gears can use that data together, determine the most optimal way to use these gears to collect the most useful data, and determine the differences in selectivity of these two gears. All right, here's a map of the sites where I've deployed my camera for these projects. I've been piggybacking off some of these larger surveys I just mentioned, mainly Alabama's Offshore Fisheries Independent Survey, CMAP, and the Greater Amberjack Count. So for the last 2.5 years, I've been going offshore as part of those surveys to deploy my cameras to collect data for those, but also collecting data for my thesis. The site selection method for these surveys is complicated, and it would take a while to explain it in detail. But essentially, I ended up with a mixture of sites between about 15 and 60 miles offshore that were randomly selected across a range of depths between about 50 and 400 feet, and also randomly selected across different types of habitat. The most common type of habitat that we got to survey is artificial reefs. Alabama has the largest artificial reef network in the country. Thousands of hard structures have been dropped offshore in order to create habitat for fish and increase the abundance of fish in the Gulf in offshore of Alabama. Each different type of reef attracts a different assemblage of fish. And these are 4 common types of artificial reefs that we survey. On the bottom are 2 types of concrete pyramids, and on the top is a metal fuel tank and a chicken coop. There's also a considerable amount of natural reefs offshore of Alabama that we survey. Natural reefs are typically rubble or rock outcrops that are covered in coral and sponges. These are the most interesting reefs to survey because they have the highest diversity of fish. Although artificial reefs attract fish, specifically recreational species like amberjack, snapper, and triggerfish, there are a lot of types of species we only see on natural structures. The most common type of habitat offshore of Alabama, though, is just plain sand bottom. We survey that too. And surprisingly, some of these videos are interesting to look at. In this video, if you look closely, you can see a flounder and a pale spotted eel swimming around. And I also wanted to show off that last year we were lucky enough to capture a great white shark on camera. And that was a really big deal for our lab because it is the first time a great white shark has been recorded in a scientific survey offshore of Alabama. So we were all really excited to be able to document that. Okay, here's what a 360 Brub video looks like compared to an ROV video. Both of these videos were taken at the same reef, just about an hour apart from each other. So because these cameras operate in different ways and have different fields of view, they have different probabilities of detecting certain species and see different numbers of fish. I'm interested in quantifying those differences, and I'm doing that by taking both of these cameras out to all of the sites that I showed you on my map, deploying the cameras one after the other, and then comparing the data between the 2 cameras. So like I said, the 360 Brub has a wider field of view than the ROV. This is important because that affects how your counts correlate to the number of fish that is actually present, because not only does that allow you to see a greater percentage of the fish community around you, but it decreases the number of time that fish swim in and out of your field of view, which is something that has a complicated effect when counting fish. The narrower field of view of the ROV means it may be more likely to miss or underrepresent something that is directly around it. However, one advantage of the ROV is that it is a mobile camera system. It has propellers, and it also has a cord that connects to the surface so we can live stream the video and see where we're going underwater. That allows us to get closer to the structures we're trying to survey and see them in more detail. The 360 Brub, when we deploy that, we throw it off the boat. We have the geographic coordinates of the reef we're trying to survey, but it's hard to get it into position because there's current, there's waves, and sometimes we're in 350 feet of water. So getting it to land close to what we're trying to look at is a problem, and that's a limitation of the gear. Another important difference is the way that these 2 camera systems move in the water. The ROV swims laterally in the water column, It has propellers, so it can swim around. That may mimic a predator. It also, the propellers make noise, so it could attract or scare certain types of fish. The 360 bruv sinks vertically in the water column, and the movement of the gear actually attracts a lot of types of fish. In this video, you can see a school of red snapper. When they see the 360 bruv sinking, they swarm towards the camera. The 360 bruv also hits the bottom. It kicks up sediment, which reduces visibility. And that also stirs up the benthic zone, which makes a lot of fish start swimming around instead of just staying on the bottom. So the first phase of my project is going offshore and collecting all these videos. Second phase is analyzing all these videos in the lab, which takes a very long time. And I'm waiting for the day when AI can handle this, but it's not quite there yet. So the first stage of video analysis is going through the videos and identifying every single species of fish that we can see. The second stage is counting the number of each type of fish that we can see. And that's a complicated process because it's hard to determine how many fish are in a video when they're moving around and they're swimming in and out of view. I'm trying to determine whether the max sends for my two gears are correlated and the average difference between the max sends for my two gears. So in order to do that, I perform regression using a generalized linear model that is particularly useful or appropriate for count data, and a prediction interval. I did remove two extreme outliers from this plot to make it look a little bit more reasonable. But what I can do with this plot is estimate what my max N for one gear should be based on the results of the other. So I can take a max N for the ROV and say, well, if I had done that survey with the 360 brub, what would I have got? So for example, let's say I did a survey with the ROV and I got a max N of 25. I can predict that had I used the 360 brub, on average, I would get 57, and then 90% of the time it would be between 196 and 14. One important difference between the 360 brub and the ROV that I have not mentioned yet is that normally the 360 brub is baited in surveys in the Gulf of Mexico. They strap a bag of squid and mackerel to the camera. And obviously bait attracts fish. So that should be affecting the relationship between my two gears. And that is something that I want to test. And my hypothesis is bait having an effect on the relationship between these two cameras? I tested that hypothesis with my preliminary data for red snapper using multiple regression. And surprisingly, it got a pretty high p-value of 0.7757, indicating that so far, there's not evidence that bait is having a strong effect on the relationship between my two gears, which was a little surprising because I've fished for red snapper in the Gulf of Mexico, and squid and mackerel works pretty well as bait. So I thought surely strapping it to the camera is going to change the relationship between my two gears. But I think the reason I'm not seeing a significant effect is because, as I showed in the video on a previous slide, when the brub sinks in the water, red snapper are strongly attracted to the camera. They swarm the camera. And so bait is not actually drawing in that many more fish than I would already see just because of the attraction to the movement of the gear. However, it's going to be interesting to test this hypothesis with other species which exhibit different behavior, like great triggerfish or greater amberjack. But at least for red snapper, because I'm not seeing a difference in the relationship, I can conclude that adding or taking away bait is not going to affect the strength of my correlation or ability to predict the results of one gear based on the results of the other. However, there are other factors related to my methods that do affect the relationship between my counts. And one of those is the analyzation period of my video. I put some plots up here showing the effect it has on greater amberjack counts. So in most surveys using brubs in the Gulf of Mexico, they turn a switch on at the surface, which starts recording a 30-minute video. Then they throw the brub in the water, it sinks and lands on the bottom, and they leave it there until that 30-minute recording is over. And when they bring it back to the lab, they only analyze the last 20 minutes of that video. The reason is because the first 10 minutes includes the part where it's recording on the boat, sinking, hitting the bottom, and kicking up the sediment. So they only analyze the part after the sediment has settled. However, that first 10 minutes is often when the highest abundance for a lot of species occurs. And so I can show with greater amberjack, there's a very significant difference between the relationship between the ROV and the 360 BRUV if you analyze the full video as opposed to only the last 20 minutes. And not only is there a significant difference in the relationship, I can use bootstrapping to generate a p-value to tell if my correlation is significantly better if I change the analysis period of my video. And with Amberjack, I can conclude that I actually have much stronger correlation between my two gears if I analyze the full video as opposed to only the last 20 minutes. This is actually pretty well known, and it's why for the greater Amberjack count, we did analyze the full video as opposed to only the last 20 minutes. But again, I can test this hypothesis with other types of species and see whether my data would be more comparable for them also if I extended my analyzation period. Okay. Now, this slide is about my species composition list. So this doesn't have to do with the number of fish that I counted. This is just about whether I saw a fish in a video or whether I did not see a fish in my video. I haven't collected counts for every single species because that takes a long time. So I only have presence absence data for some fish, but I can tell from my presence absence data whether one camera has a statistically higher probability of sighting a certain fish, certain type of fish than the other camera. So 4 types of fish I know that the ROV has a statistically higher probability of sighting are lionfish, soapfish, damselfish, and vermilion snapper. All of these types of fish, with the exception of the vermilion snapper, are cryptic, smaller species of fish. They like to stay close to reefs. They like to hide within the structure. And the ROV probably has a higher probability of sighting them because it's mobile. It can get closer to the reef and see the reef more in detail. The vermilion snapper exhibit a behavior that is kind of the opposite of red snapper. When they see the BRUV sinking, they tend to run away instead of swarm the camera. That's probably why the ROV has a higher probability of sighting those. The BRUV has a higher probability of sighting filefish, sharks, mackerels, and flounder. All of these, with the exception of the flounder, swim further away from the reef, higher in the water column. The BRUV probably has a higher probability of sighting those because of the wider field of view. And the flounder also probably has to do with the field of view, but also the fact that the BRUV picks up sediment. It disturbs the benthic zone, which causes them to swim around as opposed to just staying camouflaged on the bottom. I've also started experimenting with cumulative distribution plots. Cumulative distribution plots show the percentage of my data that occur within a particular interval. So each dot on this plot represents a site where I collected a max N for the red snapper, and the time is the length of time that the camera was on the bottom before I collected that max N. So if I have a x-axis value of 5 minutes, that means the camera was on the bottom for 5 minutes before I collected my red snapper max N. This is important because it allows me to see how long I need to record in order to collect useful data, which is important for anyone deciding how long they want to soak their gears, but also important because 360 BRUV surveys and ROV surveys typically last different amounts of time. An average BRUV survey spends about 26 minutes and 40 seconds on the bottom of the ocean. An average ROV survey is about 10 minutes and 11 seconds long. So the difference in the recording durations of these two videos is affecting the difference between the number of fish that I'm counting. And I can tell using my cumulative distribution plot that only 77% of the time, the max N for the 360 BRUV is happening within the same duration period as an ROV records for. And I can test this, how this is affecting other types of species. It's less relevant for greater amberjack, but still a factor. I can also fit a logarithmic function to this plot and extrapolate, and that allows me to picture how my max Ns would change if I left my gear on the bottom for a longer period of time. So let's say I left the gear on the bottom for an hour instead of just 26 minutes. I can estimate how that would change my greater amberjack max Ns, and I can say there's probably around a 6.55% chance that I would actually get a higher max N if I left my gear on the bottom for longer, for an hour, which is not a very high chance. That's just about 1 in 15. So earlier in the year, we were working on a project and some statisticians asked me like, well, are we gonna see more fish if we just leave the gear on the bottom for longer? And for greater amberjack, I can say it's pretty low probability. So I can also test that with other species. I can also use cumulative distribution plots for other variables. I could estimate how species richness would change if I left the gear on the bottom for a longer period of time, or just how the probability of sighting a single species changes if I leave the gear on the bottom for longer. So that's all the general ideas behind my hypotheses. I'm thinking about maybe doing a couple other things, maybe a t-test to compare species richness, or trying different models to see if they fit my max N trends better. But in conclusion, both of these two gears are important tools for fisheries managers. And each one is surveying the same habitat, but they're telling a little bit of a different story of what is there. And it's important to compare and correlate this data to make that more usable and applicable for fisheries managers. So I'd like to thank my advisor, Sean Powers, and everyone in the Powers Lab, especially Crystal, Mark, and Kai, who I could not survive without, and also the crew of the Escape, who we do most of our field work, and my parents, who I think are on Zoom, who probably don't hear from me enough. But other than that, yeah, I'd like to open up for questions. Yeah. Mhm. What? Sorry. A logarithmic function is what I fit to the curve. Yeah. Um, I don't necessarily understand the question, but yeah. Right. Yes. Yeah. 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 Well, the I'm not showing the number of fish I'm counting on that plot. That's not related to the plot. That is just oops, the point in the video when the max N is occurring. Yeah. So mhm. Yeah. So yeah, so the cumulative distribution plots are not related to the number of fish I'm counting. It is just the point in the video when I'm collecting that data. So it is not related to how much fish there are there. Okay. Yeah. Mhm. Uh-huh. Right. Yeah. Right. So visibility is definitely a factor that affects the cameras. Um, so there is less light offshore, but something that affects visibility more is just the turbidity of the water. Sometimes you still have way less visibility near shore. Actually, you usually have less visibility near shore in shallower water than offshore because you get further away from the the rivers that are pumping all the sediment in. So we do classify our videos into, I guess, 1 to 5 on a scale of visibility. And yeah, that might be a factor that I use to compare my data when I do comparisons. So that could be another variable that I add into my plots. Yeah. Mhm. Yeah. Right. Right. Mhm. Yeah. 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 So the ROV it swims up to the reef, it spends a few minutes on one side, then it leaves, goes around, spends a few minutes on the other side, goes to the top, and then looks down and spins. So it does get a couple different angles and also views of the area around the reef. The bruv just sits there and watches everything. So yeah, I would say the reason why the bruv's probably seeing a lot of species is because it spends way more time looking around. But the ROV does get some views of what's in the surrounding area too. Yeah. Mhm. Mhm. Yeah. 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 I mean, that would fix the problem of the bruv not landing next to the reef. Yeah. We could make one. These things are just standardized gears. that are the state and the federal government has decided like this is what we're going to use, and repeating that over space and time is very important. But I mean, if I could make one, I'd put a 360 bruv on the ROV if I could decide what should be used. Yeah, yeah. Mm, yeah. Mhm. Mhm. No, yeah. Yeah, right. Yeah, because we are comparing other gear types like sonar gear to this too. And yeah, I'm hoping that when I add more points to that plot, I get less variability. It seemed to follow a general trend, but then every maybe like 1 out of 6 videos is just way off. So yeah, so far, yeah, I'm kind of disappointed with the correlation. Um, yeah, so this is going to be tested versus other gear types. I don't know the answer to that question yet, but yeah, maybe Crystal can provide more. Oh, yeah. Oh, yeah. Mhm. Mm. Right. Yeah. Right. Yeah. Mhm. Yeah. Yeah. I haven't had time to just, yeah, a lot of the surveys, they only analyze one camera instead of the full 5. We did the full 5 for the greater amberjack count. And that is honestly, I think, just to save time. Um, yeah, so that actually would be a really important study that I haven't had time to do that, just comparing one of the 360 bruv cameras to the full 360. But I actually think that's a very important question that deserves its own study. Yeah.